Hello everyone. Today we will be doing a panel discussion on the topic centrifugation and cell fractionation. These methods has revolutionized our ability to study inner workings of cell by enabling the isolation and analysis of individual cellular components. Without further ado, Mr. Som Rigden, can you give a brief introduction and overview on it?
Oh, sure, it's about centrifugation and cell fractionation. What is centrifugation? Centrifugation is a laboratory technique that uses centrifugal force to separate components or mixture based on their size, shape, and density. The mixture is spun at high speed in a centrifuge, causing denser particles to move outward and settle at the bottom, forming a pellet, while lighter components remain in the supernatant liquid above the pellet. Now, let's understand what is cell fractionation. Cell fractionation is the process of breaking open cells and separating their internal components, organelles, to study them individually. Cell fractionation typically involves homogenization, physically breaking cells to release their contents. Centrifugation, spinning the homogenate to separate cellular structures. This allows researchers to isolate structures like nucleus, mitochondria, ribosomes, chloroplasts, and etc. Why are they important in biology and research? They are important to understand cell structure and function, enable scientists to study specific organelles in isolation to understand their roles. They are also important in biochemical analysis. Proteins, enzymes, and other molecules can be studied without interference from other cell parts. Finally, disease research allows detailed examination of disease versus healthy cells at subcellular level. Now, let's look into real-world applications. Firstly, let's see medical applications. It is used in cancer research, identifying altered organelle function, example mitochondria in cancer cells. Diagnostics, isolating viruses or bacteria from blood samples for testing. Therapies, studying drugs' effect on specific cellular components. Secondly, biotechnological applications. It is used in protein production, isolating cell machineries like ribosomes or for studying or enhancing protein synthesis. It is also used in vaccine development, purifying viral particles or components used in vaccines. And obviously in genetic engineering, extracting nuclear or DNA for modification. Finally, research and development applications. Used in organelle function studies, understanding how energy is produced, mitochondria, or how proteins are modified, Golgi approaches. Also used in metabolic pathways, analyzing enzymes in specific compartments. Lastly, it is used in environmental biology, studying microbial communities and how they respond to pollutants by separating cellular contents.
Thank you for the information. Mrs. Sombelki, can you share on the principle of centrifugation?
Thank you for your question. So, today I'll be talking about the principle of centrifugation. What are some of the principles of centrifugation? Centrifugation is based on the principle of centrifugal force and sedimentation. Centrifugal force: when a sample is spun in a centrifuge, particles experience a force that pushes them outward from the center of rotation. This force is what separates components based on their mass and density. Sedimentation: particles in a mixture settle at different rates under centrifugal force. Denser and heavier particles move faster and form a pellet at the bottom, while lighter particles remain in the supernatant. The rate of sedimentation depends on size and shape of the particles, density difference between the particles and the surrounding fluid, centrifugal force applied dependent on speed and radius. Factors affecting centrifugation: speed, RPM or RCF. Higher speeds increase the force applied, leading to faster separation. RCF, relative centrifugal force, is more accurate than RPM because it accounts for rotor size. Time. Longer spins allow more complete separation but may cause damage to delicate structures. Density of particles and medium. If the particle is denser than the medium, it sediments. If it's lighter, it floats, useful in density-based methods like gradient centrifugation. How many types of centrifugation are there? There are two types of centrifugation: differential centrifugation. Principle separates components by size and density through a series of increasing speed spins. Process. First, low speed spin belays the heaviest particles like nuclei. Next, higher speed belays medium-sized organelles like mitochondria. Final high speed isolates small particles like ribosomes. Use basic separation of organelles from cell homogenates. Density gradient centrifugation. Principle: particles move until they reach a point in a gradient. Example given: sucrose of cesium chloride, where their density equals that of the surrounding medium. There are two types of density gradient centrifugation. Rate zonal separates based on size and shape. Particles settle through the gradient before mixing. Isopycnic separates solely by density. Each particle forms a band at its own density. Use purification of DNA, RNA, viruses, and organelles with very similar sizes but different densities.
Now, we are all familiar with the principle of centrifugation, right? Mr. Tanisiri, can you please share on the cell fractionation process?
Thank you for the question. So today we're going to talk about what is cell fractionation and what are the various steps that are involved in the process of cell fractionation. Today we're going to look at a very important technique that allows scientists to study the function of organelles that is called cell fractionation. Purpose: to isolate and study individual cell organelles, example, nucleus, mitochondria, lysosomes, and ribosomes. Step 1. Tissue collection. A sample of tissue is taken, for example, heart muscle tissue containing the cells of interest. Step 2. Homogenization. Homogenization breaks open the cells to release cell organelles. It is performed using a blender or a homogenizer, which is a glass tube with plunger. Next, the buffer solution is added. This helps maintain the constant pH, which prevents the enzyme from denaturation. It also has the same water potential as the cell, which prevents osmosis and organelles from bursting. The homogenizer is then placed on ice. This slows enzyme activity to protect cell organelles. The result is a cell homogenate, a mixture of all organelles and cell components. Step 3. Cell fractionation or differential centrifugation. It uses a centrifuge to separate organelles by size and density. Centrifugation speed. At low speed spin, we get nuclei as the pellet. Moreover, we also get the supernatant, which is the remaining cell organelle. Medium speed spin. Here we get mitochondria as the pellet. High speed spin. For high speed spin, we have lysosome as the pellet. Finally, at very high speed spin, we get ribosome, which is the smallest organelles, as the pellet. Final notes. Keep pellets on ice to slow enzyme activity and protect cell organelles. Complete separation is difficult because: number 1. Fractionation may be slightly contaminated with other cell organelles. Number 2, organelles like endoplasmic reticulum and Golgi apparatus may also be present but often gets damaged during homogenization. So, hello everyone. Today we're gonna talk about how the centrifugation works. As you can see, the centrifuge is composed of many components. If you come closer, this one at the top is known as the lid. When we open the lid, we can see the rotor inside. Now this rotor holds the test tube bucket, which holds the test tube. Now when we collect the sample from the sample tissue, we put it inside the test tube and we put the test tube inside the test tube bucket. And we place the test tube holder inside the rotor. The rotor is connected to the motor, which helps it to rotate. Moreover, the space around which the motor rotates is known as the chamber. So, we do not turn on the centrifuge machine when the lid is still open. Therefore, we have to close the lid. Remember that. As you close the lid, if you can come closer once again, we can see the control panel. It can be used to set the RPM, which is the number of rounds it makes per minute, and also the timer. For this specific centrifuge machine, the RPM and the timer are already set. Now, let's talk about the principle of centrifuge. The centrifuge machine works on the principle of centrifugal force. When the rotor begins to rotate around, heavier components feel greater gravitational pull and begin to move towards the bottom of the test tube, whereas the lighter component experience less gravitational pull and remain at the top.
Basically, cell fractionation is the overall process, whereas centrifugation is a key step within cell fractionation. Mr. Somdoji, there are different types of centrifuges. Can you tell us what makes each one special?
Thank you for the question. So, each type of centrifuges are special due to a combination of factors tailored to specific needs in the lab. Different types of centrifuges. Microcentrifuges, small benchtop models for processing small volumes, example microfuge tubes. High speeds up to 13,000 to 15,000 RPM for pelleting cells, DNA, proteins. Common in molecular biology labs. Benchtop centrifuges, versatile for medium volumes, example 15 ml, 50 ml tubes. Used for various applications like cell separation, reagent preparation. Moderate speeds. High-speed centrifuges can reach higher speeds than benchtop models. Used for separating smaller particles like organelles or bacteria. Often refrigerated to prevent sample degradation. Ultracentrifuges, very high speeds, 60,000 plus RPM, regenerating very high G forces, example lesser than 100,000 gram. Used for separating very small particles like viruses, ribosomes, proteins, and nucleic acids, and for density gradient centrifugation. Require vacuum systems to reduce friction and heat. Can be analytical for studying sedimentation properties or preparative for isolating components. Clinical centrifuges, used in hospitals and clinical labs for separating blood components, plasma, red blood cells, platelets, urine analysis, etc. Typically lower speeds and designed for safety and ease of use. Their applications in research and medicine. Research. Isolating organelles for biochemical studies. Purifying DNA, RNA, and proteins. Separating cells from culture media. Fractionating cellular components for analysis. Studying sedimentation rates and molecular weights. Medicine. Separating blood components for transfusions and diagnostic tests. Concentrating cells or pathogens from patient samples. Preparing platelet-rich plasma (PRP). Analyzing urine and other bodily fluids. Example of a real lab process using a centrifuge. Isolation of mitochondria from cultured cells. Cultured cells are harvested and homogenized to break open the cell membranes. The homogenate is subjected to low-speed centrifugation to pellet large debris, example, unbroken cells, nuclei. The supernatant liquid above the pellet is collected. The supernatant is then centrifuged at a higher speed to pellet the mitochondria. The resulting mitochondrial pellet can be resuspended in a buffer for further biochemical analysis, example, enzyme, assays, protein studies. Ultracentrifugation with density gradients can be used for further purification of the isolated mitochondria.
Now, you're all informed about the different types of centrifuges. Question again goes to Mr. Tanri Siring. What are the application and future trends of centrifugation and cell fractionation?
Thank you once again for the question. So, some of the future trends and applications of centrifugation are.
Some of the applications and future trends of centrifugation in disease research are: number 1, cancer, used to isolate cancer cells from blood or tissue to study their behavior, gene expression, and drug resistance. Number 2, neurodegenerative diseases, helps isolate brain cell organelles, example, mitochondria, lysosomes, to study diseases like Parkinson's or Alzheimer's. Number 3, infectious diseases, used to purify pathogens, virus, bacteria from patient samples to better understand infection mechanisms. Centrifugation is used in vaccine production. Number 1, virus purification, key step in isolating viruses or viral components used in vaccines, example, influenza, COVID-19. Number 2, protein isolation, enables extraction of viral antigens and proteins for subunit vaccines. Number 3, cell culture processing, used in scaling up vaccine production by separating desired cells or viral particles from growth media. Introduction to advances in centrifugal technologies. Number 1, ultracentrifuges, spin at extremely high speeds over 100,000 RPM, allowing separation of very small particles like viruses, ribosomes, and exosomes. Number 2, refrigerated centrifuges, maintains low temperatures to preserve sensitive biological samples like proteins and enzymes. Number 3, automated centrifuges, integrated with robotics and sensor for high throughput labs, improving speed and consistency. Number 4, microfluidic centrifugation, miniaturized systems used in point-of-care diagnostic and lab-on-a-chip devices. Future applications in biology and medicine. Number 1, personalized medicine, rapid isolation of patient-specific cells of exosomes for targeted therapies. Number 2, gene therapy, purification of viral vectors like AAV for safe and effective gene delivery. Number 3, regenerative medicine, isolation of stem cells or cellular components for tissue engineering. Number 4, liquid biopsies, using centrifugation to isolate circulating tumor DNA or exosomes for non-invasive cancer diagnostics. Number 5, space biology, compact centrifuges for studying cell behavior in microgravity environment.
Wow, there are many implications of centrifugation and cell fractionation in the coming days. Mr. Somrigden, can you conclude la?
Oh, to conclude, centrifugation and cell fractionation is nothing but it's a technique used to study cells, diagnose disease, and advance researches. Basically, in short, it's a small device with big impact.